This is called The Secrets of the Soil Sociobiome. So I've been reading all kinds of really in-depth ecological articles about the science of how microbes communicate with each other and communicate with plants, and how plants communicate with each other. And then I thought, now how am I going to put this into something that actually translates to, something that's understandable? So I've had a go, and this is the first time I've actually given this webinar as well, The Secrets of the Soil Sociobiome. So we'll see how we go. So let's just see if I can now. So soil is incredibly biologically diverse, and it's got all kinds of critters in it. I'm sure that everybody that's watching this today recognises this fact that there are all kinds of amazing things in soil. It really is the last frontier when it comes to science. And biodiversity in all of our ecosystems, not just in our soil, is actually the key factor in the effective functioning of living systems. It doesn't matter whether we're talking about, the ocean or a rainforest or on our farms or in the soil. Biodiversity is actually the key factor we're realising more and more as we look into the science of these things. And in terms of soil biodiversity, it is so important for nutrient cycling, for moisture retention, for resistance to pests and diseases, for crop and pasture productivity, for food security, landscape function in terms of, we were just chatting before this session started about Australia's just had massive droughts and then massive floods, and it's also very important for carbon sequestration. Well, I'm not going to go into any of those things in any particular detail today. I mean, I'm sure you're all very well aware of why soil biodiversity is important. We're just going to go on and actually look a little bit more into, the secrets of the soil microbiome. But the Food and Agriculture Organization of the United States has estimated that the socioeconomic value of soil biodiversity exceeds $1,542 billion, which all I can say about that is that it's an awful lot of money. But in other words, this is very valuable. Well, the things that are going on in the soil that we really don't know very much about are very important for us. So far, so good. Soil biodiversity is important. Even the Food and Agriculture Organisation has recognised that it's worth dollars. But how do you get it? This is the practical part of the equation. How do we know that we have biodiversity in our soil? And if we don't have it, how do we get it? Well, there's lots of issues with trying to figure out what's actually going on in our soils. One of those is that less than 1% of soil microbes are culturable. And what that means is that you can take some soil and you could put it into petri dishes in a laboratory or something, and you could provide food for microbes. And less than 1% of the microbes that were in that soil would actually grow in that medium. So we can't culture them in a laboratory. We can't study them. It makes it very, very hard to know what is actually in your soil if 99% at least of the microbes there refuse to cooperate in terms of allowing us to study them. So number one, they can't be cultured in a lab, most soil microbes. And number two, the majority of microbes in the soil at any one time are actually in a non-active state. In other words, they're dormant, which again, that makes it very hard to be able to detect unless they've been activated by something. So the soil microbiome or the soil sociobiome is very difficult to study, and that difficulty has led us up several garden paths or taken us down the wrong track many times. We've had to rely on models, and we still are relying on models to some extent to try and model what is actually going on in the soil. And the models that we've used in the past haven't really been correct because we haven't had the technical skills to actually detect what was in the soil. Now, in 1987, there was a paper published which is called the Classic Soil Food Web Model. H.W. Hunt was the lead author of that, but there was quite a few authors on that paper. And that classic food web model, unfortunately, is still being used and still appears actually in a lot of USDA publications, but it was theoretical. It was based on estimates. It was basically a theoretical calculation of the nitrogen transformations that take place in the soil through what was called the detrital food web, or if you like, the decomposer pathway in the soil. In other words, you have certain organic compounds in soil, like soil organic matter, for example, and it's broken down initially by fauna in the soil, and it's sort of broken into smaller and smaller and smaller components and goes through, well, basically a food web where everything eats everything else. And nitrogen was thought to be released in the soil through that pathway. And some nitrogen definitely is released into soil through that pathway, but it is not the main pathway in soil. And I'm going to talk about nitrogen in a couple of weeks' time. There's going to be a webinar called The Nitrogen Solution, and I'll go into that in more detail. But we have become really hung up on this classical soil food web model, which in actual fact is now being shown through the advances in technology we have now is substantially revised. So there are, lots of articles about, soil food web revisited and soil food web challenged, et cetera, et cetera, et cetera. So empirical studies, in other words, where we really do have data to go by, have found that if we're looking at carbon cycling, if we're looking at nitrogen transformations in soil, that most of these take place through the fungal energy channel. So it's not through the herbivory of bacteria, for example. Some does go through that channel, but the fungal energy channel is the one that's really important. And the fungal energy channel, the main fungal energy channel, begins with labile carbon in the form of plant root exudates. In other words, we're talking about hundreds or sometimes even thousands of different carbon compounds that come out of plant roots for various reasons, sometimes to feed microbes, sometimes to signal to microbes. And how do we know that that fungal energy channel is operating? We look to see whether the roots of our plants have riser sheaths, in other words, whether they have soil sticking to the roots. And then we know that that fungal energy channel is actually open and that the plant is actually channeling energy out into the soil ecosystem. I've showed this slide many, many times, and I probably will show it many more times because one of my favourites. This is a high magnification photograph of what that actually looks like inside a riser sheath. You can see there are lots of fungal hyphae in there, and they're not necessarily symbiotic fungi. In other words, they're not necessarily mycorrhizal fungi or trichoderma or other fungi that form a very close relationship with plants. The more research that's been conducted, the more we've been able to see that a lot of them are what are called saprotrophic fungi. So they're basically just feeding off those sugars that are coming out of plants. And it has been thought under the classic food web model that fungi derive most of their energy from sort of lignaceous material or materials that are difficult to decompose, like woody materials and things that bacteria, for example, are not able to access. That's thought to have been the fungal pathway, and certainly you do see fungi decomposing those kinds of material. And then it has been thought, well, if you wanted to have more fungi in the soil, you would have to have more of those less readily decomposable type materials in your soil. Well, the science shows that, in fact, the most prominent fungal energy channel is actually labile carbon, the kind of carbon that was thought to be what bacteria feed on. It feeds enormous numbers of fungi and many, many varieties of fungi. And the more we look into it, the more we realise that these saprotrophic fungi are very, very important. And they are moving labile carbon away from plant roots out into the soil food web as well, not just using it for themselves, but also feeding bacterial colonies. And in fact, we now know that the hyphae of fungi have their own bacterial rhizosphere, if you like, in the same way that plant roots have a sort of a coating of bacteria around them, of bacteria that are actually feeding on exudates from the plant roots. The fungal hyphae that are associated with plant roots also have a coating, like a biofilm, of bacteria around them that are feeding on exudates from those from the hyphae themselves. So the hyphae exude carbon as well as the roots exuding carbon. So it's worlds within worlds. The closer we look, the more we see. And this is a lovely, I think I did show this one in the phosphorus paradox, a high magnification view of plant root with all the exudates around it. And you can see some of these droplets of exudate here are being cradled by fungal hyphae. They could be any kinds of fungi. There's a whole lot of really beneficial fungi around plant roots, unless we're using fungicides, of course. And it has been shown that fungicides, of all the poisons that you would use on plants, fungicides are the most detrimental to the soil because, of course, they are preventing this entire fungal energy channel from even getting started. And this is a very, very well-known slide. I've used it hundreds of times. Other people around the world used it hundreds of times from the Aberdeen Mycorrhiza Research Group. It's quite an old slide, but it just very neatly demonstrates how the hyphae, in this case of ectomycorrhizal fungi, can move energy that's coming from photosynthesis being channeled into the soil by plant roots and can actually distribute that. They are the highway and the internet of the soil. And we know this about mycorrhizal fungi, but we have to start thinking about lots of other kinds of fungi as well that we can't see as easily as we can see these ectomycorrhiza. This is a close-up of a plant roots coming down the centre of the photograph here, and then everything else that you can see in that photograph are the hyphae of, in this case, ectomycorrhizal fungi, but they can certainly have a wonderful network throughout the soil. So this fungal energy channel is the key pathway to support biodiversity in the soil, because these fungal hyphae are keeping most other soil organisms alive through their energy networks, and also joining plants through common mycorrhizal networks. If I have time at the end of the presentation today, I'll just give a little example of that. But messages can be transmitted from one plant to another through these hyphal networks, as well as energy, as well as water and nutrients. So the whole process of photosynthesis and plant root exudates are recognised as constituting the primary pathway for soil building. So if you're still thinking in terms of the classic soil food web model, you need to just put that one aside and think about this fungal energy channel. So here we have a plant in the centre of the diagram here with the root exudates coming down this primary pathway. The exudates do have to move through the microbial pool, and talk about microorganisms a little bit more in a while. And this is, at this stage, it's all labile carbon, but it is going to be processed through the soil microbiome, and we want it to end up as being stable soil carbon down here. And our fungi and our bacteria, in this instance, it's being called the microbial carbon pump, are very important for the transformations that take place. So over here on the left-hand side, where we have leaf litter and decomposing roots, and there might be animal manures, or there could be compost, or whatever the sorts of organic matter, actual matter itself, that's composed of lignin and cellulose and hemicellulose. That decomposition pathway does feed into the stable carbon pathway in the form of the enzymes that microbes use while they're decomposing these materials, can actually stimulate this other liquid carbon pathway, but it is peripheral to the main pathway. It's important. So here's a close-up of Scott Ravenkamp holding a plant that's an oak plant that's had lots of exudates coming out of the roots, and it's created wonderful transformation for this soil, which over here on the left-hand side, you can see that the soil in the absence of plants is very compacted and doesn't have any structure. So one of the important things about these exudates is what they can do to soil structure is a highly magnified view, actually, of some sand that's been incredibly improved through soil fungi being supported through the soil, through the liquid carbon pathway. So this has got nothing to do, this soil has had no plant residues or any organic matter added to it. The structure is just improved with fungal hyphae and the fungal energy channel. So over on the right-hand side, we have soil that's well aggregated through that pathway, and on the left-hand side, we have compacted soil where if our soil particles haven't been pulled together with fungal hyphae, then we won't have those lovely spaces between them for air and water to pass through. So we know that plant root inputs build carbon 5 to 30 times faster than carbon derived from above ground biomass. And this above ground biomass is, of course, the basis of the classical soil food web model, which has now been, let's just say, revised. So when our fungal energy channel is operating, this is a photo from Derek Axton's farm. It's a little durum wheat plant at a very young stage of growth. The seed is just here under my pointer. We have this fantastic seminal root system that's getting down really, really deep in a very short amount of time, and great riser sheaths on the crown roots here of this plant. And these plant root inputs, they're really the only way that we can build carbon at depth. We can put organic materials, like we can have our crop residues near the soil surface, or we can add compost or something at the soil surface. But if we want to build soils at depth, in this case we're looking at about 12 inches of depth, then we have to have plant roots at that depth, and we have to have plant roots that are actively exuding labile carbon for that fungal energy channel. So why have I used the term the soil sociobiome for today's webinar? Well, it's because what is actually happening in that soil that we can't see when we look at it, basically we see a whole heap of dirt, I guess, is that we can't see, unless we're looking for riser sheaths, we can't see that there's interactions taking place between plants and microbes. And there's also many interactions taking place between microbes and plants. And I will give an example of one of those right at the end, and multiple interactions taking place between microbes and microbes. There's all these different levels of things going on in the soil, and really it is a social whirl. It's unbelievable, the amount of the chemical messages, the biochemical messages and the signals that are going on. There's probably thousands of scientific articles now published on just exactly how all of that messaging works in the soil. But all of the interactions between plants and microbes, microbes and plants, and microbes, they all involve biochemical signaling. So you have to think of it in terms of these mostly single-celled things in the soil, little tiny cell, too small for us to be able to see with the naked eye. And of course, microbes can't see, they can't hear anything, they can't smell anything. I mean, they don't have any of the senses that we as a human being have to be able to detect our environment and respond to our environment. And the only thing that they can respond to temperature, they can respond to moisture, and they can respond to biochemical signaling. And the biochemical signals enable a microbe to know where it is in the soil and what it is that it needs to do, what its neighbours are, how many of them there are, and enables microbes to actually coordinate their behaviour and achieve all kinds of amazing things in the soil, like building aggregates, for example, or bringing nutrients to plants. And these biochemical signals are very complex. There are thousands of them. And it's really quite an extraordinary world. I don't know any simple way to explain it really, other than there are every biochemical signal has a different shape. It's a shape in terms of what it's made from, the atoms that it's made from, the elements that it's made from, and those shapes will fit into receptor sites, both in the microbes that generated them and in other microbes that are able to receive them. And this is exactly the same way that biochemical signaling takes place in the human body or in any living system, is that there will be biochemicals produced and organs, if you like, or cells that are able to either read those chemical signals or ignore them. And if there is a receptor site, then the signals can be read. I think I'll probably just leave it at that. But this is basically how the thousands of different chemicals are sorted in the soil as to, like, when there are so many things living in the soil and so many things, the only way that they can communicate with each other is through biochemical signals. It's just absolutely extraordinary how it all works. But when you stop and think about it, it works the same way in our bodies. In our human bodies, we have, there's hundreds of different biochemicals floating around right now that are telling your liver what to do and your kidney what to do and your heart what to do. And and it's all regulated at that level without us really having to think about it. But what we do know about plants now, with the technology that we do have and the fact that we're able to interpret some of these signals, is that plants function at their best when there is many different kinds of microbes living around their roots. And the best way to achieve that is by having many different kinds of plants growing together, because when we have them, every kind of plant is obviously supporting different kinds of microbes. And when we have the plants growing together, we want them to actually, we want root mingling. In other words, we want the roots to be mingled in the soil. They don't necessarily have to be touching each other, but the plants have to be close enough for the mycorrhizal fungi to be able to link them together in a common mycorrhizal network. And even though there are plants that are not mycorrhizal, like brassicas, for example, provided they're in a mix with other kinds of plants that are mycorrhizal, they will join into the common mycorrhizal network, although they don't contribute to it. They are still part of it, and they're still joined to the other plants through that network. So what effect does this root mingling of different plant species have on plant productivity, on immunity to pests and diseases, on tolerance to stress, like droughts or waterlogging? As I mentioned before, here in Australia, we had massive droughts, and now we're underwater, and there's houses floating away and cattle floating away, and but the things that live in the soil have to deal with all these stresses, and these things come around in a cyclical nature all around the world. That there's always various stresses, that environmental stresses that plants have to deal with and microbes have to deal with as well. So given that plants are in an environment that can be quite harsh at times, and given that we expect our plants to be productive, we want to be able to harvest them, or we want to be able to graze them, it's very important to know what effect root mingling, or the sharing of the sociobiome, actually has on those functions. So in this diagram here, I know it looks rather busy, and that's because I guess they're trying to put the plants a little bit in the background and bring the microbes to the foreground. On the left-hand side, we have a bean plant, and on the right-hand side, we have a rice plant. So one is in the Fabaceae family and one is in the Poaceae family. And to get the maximum benefit from root mingling, we really want our plants to be in different families. So when I talk about putting different kinds of plants together, I mean plants that have got different functional traits or come from different plant families. So, for example, if you wanted to put 6 different kinds of plants together, there's not a huge amount of benefit of putting 6 different grasses together. If you could put plants from 6 different plant families, then that's going to be much more beneficial. So, sorry, I just have to. These days when I'm not doing so many workshops and things, I don't get to use my voice a lot. So on the left-hand side, we have our bean plant. And you can see that there's a whole lot of microbes around the roots, and they're obviously the rhizosphere microbiome. There's also microbes all over the plant, on the leaves and the stems and the flowers and the fruits, and they're that's the phyllosphere microbiome. And then we have the endophytic microbiome, which is all the microbes that are actually inside the plant, the endophytes. And the more we look at these microbial interactions, the more we realise how important endophytes are. And most of the endophytes that are in the plant have actually come from the soil. So the plant has taken them up from the soil for some reason, for something that the plant needs. And quite often that could be to help the plant to deal with a nutritional stress of some kind, or to deal with an environmental stress of some kind. It will take from the soil sociobiome, if you like, what it needs. Now, if we have different kinds of plants growing in close enough proximity for them to be able to share those microbes, what we will find is if there's different kinds of microbes under one kind of plant that aren't under the other, or aren't in association with the other kind of plant, if they are able to mingle their roots, or if they're able to be joined by a common mycorrhizal network, they're actually able to use the genetic material from another plant. So the microbes that are living in association with plants are able to the genetic material that's in those microbes is able to be used by the plants for stress tolerance and also for the acquisition of nutrients. So plant productivity is very much influenced by diversity, by the biodiversity of the microbiome. So if we have two plants the same, for example, two corn plants or two wheat plants growing side by side, they're going to have very similar microbiomes, in fact, probably going to have identical microbiomes, and that's going to have a negative feedback effect on plant productivity. The microbiome is going to be able to detect that the microbiome that's next to it or near it is the same as itself, and therefore it is a competitor. The microbiome is what will detect that this is competition. It's not the plant detecting that there's another plant it's competing with. It's the microbiome detecting there is another microbiome that it's competing with, and it basically won't cooperate, won't share resources. If we have dissimilar microbiomes next to each other, so we have plants from different plant families next to each other, and there are 6 or 8 plant families that are quite commonly used in agriculture. There's no reason why you can't have 6 or 8 plant families together. That will have a positive feedback effect on plant productivity. And you'd wonder, well, why would that be, that the microbiome, when it detects that other microbiomes in its vicinity are dissimilar, why would it be that it will actually cooperate rather than refuse to cooperate? You know, why does it cause that change? It's a change in behaviour by the dissimilarity. The more dissimilar they are, the more they will cooperate. And this is because if we think of a diverse plant community like a native prairie or something like that, where we have lots of different kinds of plants growing together, they will have different functional traits. In other words, they will respond to different environmental stimuli. Some may grow well when it's hot, some may grow well when it's cold, some may prefer it when there's lots of soil moisture, others could be very drought tolerant. And that means that at some time of the year, almost any time of the year, there will be something that is able to grow. And if there is something in that plant community that is able to grow at most times of the year, and all the plants are connected underground by a common mycorrhizal network, as well as other networks, that's the one that we're most familiar with, but we now know that there's a lot of networking going on in the soil, then it means that there is energy coming into that microbial network continuously year-round. If you only have one kind of plant there, it's only going to grow productively at one time of the year or under one set of environmental conditions. And for the rest of the year, the microbes in the soil basically starve. So it is to the benefit of the soil microbial community to have as many different kinds of plants there as possible. And the microbial community at some level is able to detect that. And if the microbial community detects that there is a lot of difference between the plants, in other words, the plants have dissimilar microbiomes, they're using dissimilar chemical messages, then the microbes cooperate and they have a positive feedback effect and they actually support each other. And this cooperation between the microbiomes, not between the plants microbiomes, actually has an effect on plant productivity. Just jumped over a slide and I'm not really sure how to go back. I'm not having much luck with my techniques today. So I have mentioned this Yena biodiversity experiment many, many times before because it's probably the classic biodiversity experiment. It's run for at least 15 years in Eastern Germany. And in this experiment, they had 4 functional groups of plants. So they had grasses, legumes, tall herbs, and short herbs. So there's 2 different kinds of non-leguminous herbs. I think it's really important to get away from just grasses and legumes when we're looking at our mixes. And they had 1, 2, 4, 8 or 16 different plant species, but remember that's 4 functional groups. So if they had 16 plants, well, let's say if they had 8 plants, they didn't just have 8 different grasses or 8 different legumes. there was 4 functional groups in there. So there would have been 2 species from each of 4 functional groups. And I think it's the 4 functional groups that's more important than the 8 species. And they looked at biomass production and beneficial insects and microbial activity and water balance and carbon and nitrogen and phosphorus. And there's been hundreds of papers written out of this experiment. But this is just an overview of the site. You'll notice that there's a river, the Yana River, just running around the back of this experimental area here, and I'll mention why that's important in a minute. But what they found is that when they had, as the numbers of species went up in their experiments, that they actually had a linear response in terms of plant biomass. So this is 1 species, 2 species, 4, 8 or 16. And remember, where you've got 4 species, they're from 4 different plant families. Where you've got 8, they're from 4 different functional groups, not just 8 of the one kind of thing. And we've got this linear response. So the more and more diversity, the more functional diversity we have, the greater the biomass. And that's going to be really important for a whole lot of reasons. Doesn't matter whether it's crop biomass or whether it's pasture biomass, or even if you're just growing cover crop for soil protection or for feeding the soil microbiome. The more biomass you have, the more photosynthesis you're going to have, and the more that fungal energy channel is going to open. And in fact, it has been shown that there is a direct relationship between plant diversity and soil fungi. So forget all the stuff about fungal to bacterial ratios of 1-to-1 or whatever people talk about. You actually want them to be a lot more than 1-to-1. And the way to open that fungal energy channel is through plant diversity. So over here on the left, we have just plants of 2 different functional groups. So it could be a grass and a legume, for example. You will only ever be able to build soil to a certain depth with that limited amount of diversity. Over here, where we have 4 functional groups and 8 species, they're able to build, this community is able to build much deeper soil because these microbiomes are actually cooperating. And this deeper soil proved to be very beneficial when that river did flood and the whole experiment went underwater. And it was sitting in water for, there's several feet of water there. It was sitting in there for several weeks. And the scientists thought that the entire experiment was going to be destroyed. But they found that the mixes that had the high diversity survived perfectly well, the waterlogging, whereas the monocultures didn't. And the other side of that coin is that drought tolerance is much, much higher, where we have high diversity in terms of a lot of different functional groups. Again, your 4 functional groups. So what happens when plants are in diverse communities is that they can respond to the drought by recruiting microbes from the microbiomes of dissimilar plants. So this is where your dissimilarity becomes really, really important in extremes of, in climatic extremes. This is where diversity really comes to the fore. Because, for example, in a drought situation, you may have a plant that, a species that of its own is not really very drought tolerant, but if it's growing next door to something that does have better drought tolerance, it's able to recruit microbes from beneath that other plant. And when those microbes come as endophytes, they're taken up through the roots and they move into the plant. They're actually living inside the plant, so the plant has to feed them. So it means it has to photosynthesize more and produce more energy to actually, like, these have become borders or lodges, if you like, within the plant, and the plant is feeding those microbes. It's not going to do that unless it has a benefit from them being there. And those microbes will be able to bring about physiological changes within that plant that the plant's not able to bring about itself. For example, just produce enzymes or something that might thicken cell walls, reduce the amount of moisture that's lost through transpiration and those kinds of benefits. There's a lot of literature on this, on how endophytes, beneficial endophytes, can actually provide drought tolerance to plants. So it's not a genetic thing, it's a microbial thing. And it comes, again, through diversity, through the sharing of this rhizosphere microbiome, and we need to have root mingling for that to happen. So the higher productivity that we see in diverse plant communities is due to a whole lot of different factors. As Noah mentioned before, it's like a jigsaw puzzle. We need to put all the different things together. You can't say it's just any one thing. For a start, if we have lots of different families of plants, then we're going to have variation in leaf architecture. Some things like the grasses, for example, are just going to have fairly narrow leaves sitting at, say, a 45-degree angle that really don't catch a lot of light. Other things like sunflowers are going to have, big wide open leaves that do collect a lot of light. So this variation in leaf architecture results in more light interception, and that means that photosynthesis is going to be proceeding at a higher rate. We have a higher photosynthetic capacity, which means there is going to be more carbon exuded into the soil, and more exudates means more microbes, and more microbes is access to a larger gene pool. So when I say more microbes, I mean more different kinds of microbes, because we have different kinds of plants. And then that's very important for soil structure, for aggregate formation, because different kinds of bacteria and fungi and plant roots and plant root exudates and everything work together in a coordinated way to actually form aggregates. Nitrogen fixing, which I'm going to talk about. I'm talking about free living nitrogen fixing, and I'm going to talk about that in more detail in a couple of weeks' time. Disease resistance, frost drought, and flood tolerance. All of these things come through microbial diversity, which in turn comes through plant community diversity. And these diverse systems are self-organising. So the microbes actually know what to do. And they know what to do because they have such complex signalling mechanisms that we're only just starting to figure out actually how this signalling works and how microbes produce these different signals, how they respond to them, how they know which ones to respond to. So, yes, it really is a very, very complex world under our feet, but it is a very, very well-organised world. So just to give you one example of, I was saying that there's a whole lot of different levels of interaction. There's the interactions that we're most commonly, that are most commonly spoken about, or that we've been aware of for the longest time, have been the interactions between plants and microbes. We know that plants produce signals that stimulate various microbes, and they stimulate microbes that they need for certain purposes. Microbes also produce signals that can have either positive or negative effects on plants. So it can work both ways. And microbes also produce signals that can have positive or negative effects on other microbes. So we have all these levels of conversation going on. And I just wanted to give you one example. I think I may have mentioned this before in other webinars, but I haven't spoken about it recently. But it's the networking that goes on between trees in a forest, which is often attributed to the trees themselves, but it has nothing to do with the trees really, other than the fact that they're photosynthesizing, and it has everything to do with the common mycorrhizal network. So this is one example here of, we have a paper birch on the left-hand side and a Douglas fir on the right-hand side. The Douglas fir, you'd be more familiar with these trees than I am, these Northern Hemisphere trees, but the Douglas fir is going to remain, it's evergreen. It's going to be able to photosynthesize at various levels throughout the year. Over on the left-hand side, we have a paper birch, which is going to lose its leaves in winter. Now, these trees are joined underground by a common mycorrhizal network, and the fungal network in the soil is receiving all of its energy from photosynthesis. So in summertime, when the paper birch has got leaves that capture a lot more light than the Douglas fir, most of the energy coming into the system is actually going to be from the paper birch. But the fungal network is channeling energy to this, in this case, a seedling of a Douglas fir, which is being shadowed by the paper birch in the summertime when it has leaves. So the seedling of the Douglas fir wouldn't be able to survive in the summertime because there wouldn't be enough light unless the fungal network kept it alive. And this is often attributed to the tree actually being very benign and keeping another tree of another species alive. But it is the fungal network that is keeping a tree of another species alive. So why should the fungal network care whether a Douglas fir seedling survived or not? Well, it's going to care because in wintertime, when the paper birch doesn't have any leaves and it's not photosynthesizing, the only energy that can come into that common mycorrhizal network is going to be from Douglas fir, which are photosynthesizing over winter. So it's a very simple example of just two species, but to show you the incredible power of the common mycorrhizal network and why the common mycorrhizal network actually behaves in that way, because it is to the benefit of the microbes to keep as many different kinds of plants alive, because that is going to ensure that there's going to be energy coming into the system for as many parts of the year as possible. So when you ask the question, who is directing traffic here, it is actually the microbes that are controlling what's going on, even though we look at those trees, like absolutely huge trees, and the energy that's moving around in the soil under those trees is being directed by microbes. So that was really all I wanted to say about the soil sociobiome, because the chemistry of it can get incredibly complex. I guess if we look at things like insects, we probably see that there's extraordinary chemical signalling going on between insects and plants and insects and other insects. I mean, isn't it amazing how pollinators they can go to a flower and then they can go back and tell others where the flower is, and then they can all go find their way back, even though it could be miles away. I mean, the signalling that we see in other ecosystems is just as extraordinary as the signalling that goes on in the soil, but we can understand it a bit better when it's something big that we can see, and we understand that it's very complex, but it is also extremely well organised. So in the soil, it is also very complex, but extremely well organised, and in the same way that insects and plants seem to be able to manage everything above ground, microbes and plants, and there's also, of course, fauna in the soil. All our invertebrates in the soil manage to get themselves organised, and most of it is through some kind of biochemical signalling. So that was all I really wanted to